Yes, so good afternoon. So you can hear me well, because me, I couldn't hear you well when the presentation was going on. So I hope it's okay.
Yeah, we can hear you well. We had issues with the previous presentation,
Okay.
but we can hear you well now.
Okay, perfect. So I think I couldn't hear a lot, but I think you already introduced me. So maybe I will skip that, just maybe to present you as the people working at the research lab. And so I think last year you had a presentation of Jérémy Courreau. So I hope I will not be too redundant with what he presented already. And so Jérémy and Cyril that were previously in the lab, and we have now other people in the lab who are still working with Dirk-Wanné syndrome, and you will show it in the slide. And also, just to introduce you, my three colleagues in France, which are the experts of Dirk-Wanné syndrome. So one is in Strasbourg, Dr. Benjamin Durand, and I have also two colleagues in the west and the south of France, Marie Vincent and Marjolaine Willems, who are like following patients in this different area, and we are really following several families with Dirk-Wanné syndrome. So just a brief introduction, but I will go quickly on that because you know everything about it. But just to show you that there are like more than 1,800 genes that are now identified. We know that they are leading to rare genetic form of neurodevelopmental disorder when they are mutated. So it could encompass intellectual developmental disorder, autism spectrum disorder, or developmental epilepsy. Most of them are involved in autosomal dominant form of neurodevelopmental disorder, and one of those genes is Dirk-Wanné, which is one of the top 20 genes, meaning that we can find it mutated in between 0.1 and 0.5% of the patient with either intellectual developmental disorder or autism spectrum disorder. All the mutations identified so far occur de novo and affect only one copy of Dirk-Wanné on the two copies we all have. And so in the team, we are working on neurodevelopmental disorder. So we are not working only on Dirk-Wanné, on different rare genetic form of neurodevelopmental disorder, but Dirk-Wanné is a kind of rare genetic form we are following since now more than 10 or 15 years, I cannot even remember. And for Dirk-Wanné syndrome, as for other neurodevelopmental disorder syndrome, our goal is to try to better describe the clinical manifestation, to improve the specific care of the patient, to develop diagnostic tool to improve the variant interpretation, because we will see after that it's not always easy to interpret the effect of variant in neurodevelopmental disorder gene, and especially among them Dirk-Wanné. We will see that we have some atypical variants for which we need more functional data to conclude about their involvement in the disease. And we also want to study a bit further how the consequence of the variant, and notably how this variant could affect brain development and then leading to a neurodevelopmental disorder. The tool we are using, so we are collecting clinical manifestation, and I will show you a different way of collection we have. We study the variant by just studying the mutant protein on using regular cell, and just to see if the variant can affect and how they affect the function of the gene we are studying, and in this case, Dirk-Wanné. And we are also using a bit more relevant cellular model, which is human induced pluripotent stem cell, which can be differentiated in 2D, into neural stem cell, and then into neurons, differentiated neuron, but also in 3D into brain organoids. And we are doing that for Dirk-Wanné syndrome, trying to see how mutations that will be introduced in iPSC could affect brain development, at least this in vitro model of brain development. So with the clinical characterization, what we are usually doing is like recording clinical information directly from clinicians, so in France, but also all over the world. But we are also, we did, for instance, for Dirkwane specifically, we did a particular research program with Dr. Benjamin Durand, who performed parental interviews with parents of children with Dirkwane syndrome. And the idea was to better characterize the neurocognitive and neurobehavioral profile of these children. And also, so the third way we have to analyze clinical data for neurodevelopmental disorder, and especially Dirkwane syndrome, is via a project which is developed in the lab by Jean-Louis Mandel, which is a GENIDA project. So it's a website where a family can connect, create a login and enter clinical information about their relative. So it could be a child or another relative in their family. And so it's something that they really can say exactly. So there are really a lot of questionnaires they can answer, trying to really recapitulate of the different aspects and how the rare genetic disease could have an impact in the life of the patient and their family. And so the idea was to do that directly with the family, because like clinician can lack of time, and so it will be easier to have directly this information from family. And sometimes family can also mention some important things that are not seen by the clinician, for instance. So you can see that in the GENIDA cohort, so now they have more than 1,000 of individuals with rare genetic form of neurodevelopmental disorder. And among them, the cohort of Dirkwane syndrome is a bit smaller. It's 38 families which have filled this questionnaire. Most of them are coming from France, but you can see that there is also 8 families from US and 7 from United Kingdom, and the other are from different other countries. What is interesting is that we have mostly children in this cohort. So, but you can see here that so the mean is 70 years old. So it means that we have also teenagers in this cohort, and also some adults that also for which the questionnaire was also filled, which can allow us to see a bit the natural history and how the Dirkwane syndrome can evolve during time. I wanted just to illustrate a few things that we can get from this website and from the parental interview. So the first one was the question of the autism diagnostic in patients with Dirkwane syndrome, who all present intellectual developmental disorder, but autism, it was not all the patients that were diagnosed with autism. You can see here in the GENIDA cohort that if we take in consideration only the family who answer yes or no, there is 42% of the people affected with Dirkwane syndrome who reach a diagnosis of autism spectrum disorder. When Benjamin Durand did his parental interview, so it was in a small number, it was 14, but so the diagnostic yield was 57%. And when we look at the literature, it's a bit different between the study. I think maybe the cohort of patients and the recruitment are not totally homogeneous, and even like the test. So in this test, we did the autism diagnostic interview revised to evaluate autism, but there are also other tests that exist. And so in the literature, the yield of autism is between 30% and 73%. So it's a bit of variable. Concerning the epilepsy, you can see in GENIDA that 75% of the family describe an history of epilepsy. So in most of the cases, is febrile seizures, and that will need a medication in 74% of the case with the epilepsy described, but none of them were drug resistant. And concerning the languages, so it reports that 1/3 of the patient has no language at all, and in 23% of the patient, it's a language but difficult to understand. Sometimes it's just word only or partial sentences, and only 10% of the patient with Dirkwane syndrome seems to do correct sentences. But it's something that could be biased by the fact that we have a lot of children in the cohort, and it's something that is really is noticed that the language can improve really after infancy. We still have during adolescence, for instance, an improvement of the language. I remember a family in France where their child began to really develop languages after 14 years old, 14, not 40. And so it could still improve after the childhood. So it's difficult to really have a view. We should see, for instance, we should try to do that with only adults with the Dirk-Wanné syndrome to really see how the language is. The other clinical signs which are recurrently associated with Dirk-Wanné syndrome are listed there, so I will not detail all of them, but for instance, there are some digestive problems in really a lot of the patients, 72%, which could be constipation, reflux, and there will be a specific questionnaire in GENIDA to really address this question more specifically and to better define this digestive impairment in neurodevelopmental disorders. Sleeping disorder in half of the patient, movement disorder also, and we are trying with some neurologists to better characterize this movement disorder in Dirk-Wanné syndrome. And so other clinical manifestation, I will not detail all of them. Some oral buccal manifestation in 1/3 of the patient with sometimes so difficult with swallowing, sometimes excessive salivation. And we can also report, and it was not reported at the beginning of the description of the syndrome, that there are also some skin and hair problem, and you can see that there are several families which report severe eczema or psoriasis in patients. So after this clinical characterization, we want to see, and sometimes it's a bit linked also, what are really the effect of variants in Dirk-Wanné. And so you will see that depending on the type of variant, it could have different effects, and it will also lead to different clinical manifestation. So Dirk-Wanné, you all know Dirk-Wanné is a kinase protein, so it means that it's a protein that can add a phosphate group to other protein and then regulate their activity. And first, before doing that, Dirk-Wanné needs to autophosphorylate itself to be able to phosphorylate the other target. And so this autophosphorylation site is located here in the protein. In red, it's the kinase domain. And so it's really the first and important essential step to be able to then phosphorylate additional protein. And so we know that Dirk-Wanné phosphorylate really a large amount of protein involved in really different cellular function, which sometimes make difficult to really understand what are the mechanisms that will lead to neurodevelopment or would affect brain development among all these targets, who are really the essential targets of Dirk-Wanné. Concerning the molecular spectrum of Dirk-Wanné syndrome, so there are now several hundred variants which have been described. So it could be structural variants, for instance, gene deletion, so deletion of the entire gene, but also variant, what we call truncating variant or missense variant. So the deletion will remove the protein made by one copy of the gene. So you will have finally half of the protein you need normally, and so it will directly have an effect because on all the cellular function of Dirk-Wanné, because you have only half of the protein and it's not sufficient to do the function. The other type of variant, which are the truncated variants, usually they introduce a stop codon in Dirk-Wanné, and so you will remove one part of the protein. So you can see that different truncated variants here that are located here and will remove really a big part of the protein. And for this truncated variant, it's quite clear that Dirk-Wanné will not be able to do its function, so we can really consider them as loss of function. The problem is the missense variant. So a missense variant is a change of only one amino acid in the entire protein. So all the rest is the same, but there is one amino acid that will change, and it's not always easy to know if this change will affect or not the function of the protein. And so we have most of these missense variants remain what we call variant of uncertain significance. It means that we found an interesting variant in Dirk-Wanné, but we cannot conclude if this variant is responsible or not of the neurodevelopmental disorder. So we cannot conclude if the individual has Dirk-Wanné syndrome or not. And so that's why we need to do sometimes functional tests to really see the effect or analyze the effect of the variant. And it's what Jérémy did during his PhD. He took some missense variant we have identified, and he tried to see how they could affect the kinase activity of Dirk-Wanné, and especially how they could affects the autophosphorylation ability of DIRC1A. So the ability of DIRC1A to put a phosphate on its tyrosine 321, which is the essential step to be able to be activated and able to regulate the other protein. So we put the sequence of DIRC1A in regular cells and the sequence without or with the mutation, and we analyze the amount of DIRC1A and the amount of DIRC1A in its autophosphorylated form here. So you can see that in the two conditions here, you have DIRC1A protein and you have also the phosphorylated form of DIRC1A protein. But in the two last condition, you have the DIRC1A protein, but you don't have the phosphorylated form of DIRC1A protein.
Yeah, you
So what we did is we test different missense variants. For instance, we test the first missense variant, which was found in the general population. So this variant, alanine 341 serine, is not predicted to affect DIRC1A function because a lot of individuals carry this variant in the general population. And so as expected, we could see that this variant do not affect the ability of DIRC1A to autophosphorylate it. But for other variants here, most
Emily,
of them, yes.
Do you mind go back a couple of slides, because here in the main room we lost a couple of slides.
Ah,
sorry.
Where did you stop?
Yes, there.
Yeah. Okay.
Yeah.
Okay. So here you can just see DIRC1A protein, and it's a position where DIRC1A will have the phosphate group on itself, which is the essential step to be able to phosphorylate the other protein. And so we will take advantage of this autophosphorylation activity to try to measure how a variant could impact DIRC1A function, trying to because we have some antibody against DIRC1A protein, but also against this phosphorylated form exclusively of DIRC1A protein. And so I was saying that maybe I can skip that just to show, because I already say it, even if without the picture should be okay, to try to describe how for some variants we can easily know that they will affect DIRC1A function, because it's the total deletion of the gene, for instance, or because it's the premature stop codon that will remove a big part of the protein. But from some variants that will affect just one specific amino acid, so you can see that the protein is more than 700 amino acids in total. So if one change, it could do nothing, or it could really affect the kinase activity of DIRC1A. And so if we cannot, sometimes we cannot conclude just looking at the variant itself, the change itself. And so, so the variant will be what we call of uncertain significance. And it's why we need to develop some functional tests to see the effect of the variant on DIRC1A activity. And so it was the work of Jeremy. So I show here, so because you couldn't see, so you can see here it's a detection of proteins that we overexpress in regular cells. So we put DNA mutant or Y-type sequence in regular cell, and we took the protein, and you can see here the regular DIRC1A, which is detected. And here, the level below, it's the phosphorylated form of DIRC1A. So you can see that in the two last conditions, you can see DIRC1A protein, but they are not autophosphorylated anymore, meaning that there is a loss of DIRC1A activity, kinase activity. And so we were able to show that different variants in the kinase and even outside of the kinase domain really impact DIRC1A activity and lead to a loss of function effects. And in this case, it was possible to consider them as pathogenic and responsible for DIRC1A syndrome. In parallel, we established, in collaboration with the Canadian team of Rosanna Wexberg, an episignature for DIRC1A syndrome. So maybe you know that for some of the neurodevelopmental disorder, it was observed that when you take blood DNA and you analyze not the sequence of the DNA, but the mark over the DNA, what we call epigenetic marks, so it's a methylation of DNA, they could observe that for some patients affected with a specific form, genetic form of neurodevelopmental disorder, the patient mutated for the same gene will share a common profile of this methylation mark. And so we wanted to see if it was the case also for DIRC1A, and it was the case. So in individuals with DIRC1A syndrome, we found more than 400 regions for which the profile is different between patients with DIRC1A syndrome and other individual, control individual. And so this could be really discriminative, meaning that if now you have a missense variant in the gene, we found a total correlation between this epigenetic signature and what was found when we analyze the effect of the variant on the kinase activity of DYRK1A, meaning that it was really robust. And so now for the missense variant we identified, we are not obliged to test the variant at the functional level, so meaning to overexpress them in regular cells, but we could directly take the DNA, analyze the methylation, and see if they have a methylation profile close to the other patient with DYRK1A syndrome. And in this case, you can consider the mutation as pathogenic, deleterious, or if they will have a profile similar to control individual, meaning that maybe this variant in DYRK1A is not causing a loss of function of DYRK1A. Another way to represent this episignature is this principal component analysis representation, where you can see here the epigenetic profile of the control, which are clustered together, and we have the epigenetic profile of the patient with loss of function variant in DYRK1A, which has also clusterized together. So now you can see for a variant of uncertain significance if the methylation signature in the patient corresponds to this cluster or this cluster more easily. But in addition to this loss of function variant, which are now well described, there are like a lot, we identified also some atypical variants in DYRK1A, and the first one which were identified is this missense variant at the end of the kinase domain, GLI486 ASP. It was identified occurring de novo in a patient, but the patient did not show the regular clinical feature of DYRK1A syndrome. For instance, he has autism and macrocephaly, so an increased head size, while the patient with DYRK1A syndrome are more reduced head size. So it was a bit different phenotype. And when we look at the autophosphorylation, the effect on the autophosphorylation of DYRK1A for this variant, we could not find any effect. But our test was discriminative for the loss of function, so we could see a decrease of the phosphorylation of DYRK1A, but it was not discriminative for the gain of function, because all the DYRK1A, when they are made, they autophosphorylate themselves directly. And so you cannot see an increase of activity with this test. But it was done by some colleagues in Spain, so they published also a test for different variants, and this time they look directly at DYRK1A activity, and you can see that this variant here, compared to the wild type, show an increase of DYRK1A activity. So it really suggests that this variant could have a gain of function effect. And when we look at the methylation signature profile in this patient carrying this variant, what we saw is that the methylation profile was totally different from the control, but also totally different from the patient with loss of function, and at the opposite compared to this patient. And so it was really a mirror episignature or methylation profile that would suggest, which is consistent with the fact that this variant could have a gain of function effect. So since this time, we have identified additional variants, or additional missense variants in or outside of the kinase domain, but also truncated variant located at the end of the protein. We show that when we remove the end of the protein, you will not affect the kinase activity of DYRK1A. You will not decrease it, actually. But it's a bit complicated, so it means that variant at the C-terminal that will lead to premature truncation at the C-terminal of DYRK1A plus some missense variants could lead to a mirror phenotype, so a phenotype different from DYRK1A syndrome, which is a neurodevelopmental disorder form, but which is not DYRK1A syndrome. And it's a bit difficult to really make the split just looking at the variant between the ones that will lead to DYRK1A syndrome and the ones that will lead to this other neurodevelopmental form, because, for instance, we have also additional truncating variant at the end of the protein that will lead to DYRK1A syndrome. And this because some variants will lead to protein aggregation, and so the DYRK1A syndrome does not lose its kinase activity, but will aggregate, so will not be able to regulate the other protein. And for the missense variant, is also the case. For instance, we have a missense variant at the same position than another missense variant, and one is leading to a loss of function, and this one will lead to a gain of function. So it's a bit complicated, and we really need to have a better overview of all the possibility, all the impact of all these different variants in Dirk-Wanné. And so in the lab, we are trying to describe this novel neurodevelopmental disorder associated with Dirk-Wanné variant, which is different from Dirk-Wanné syndrome. So with variant having a gain of function or a different effect than a loss of function in Dirk-Wanné. What we can see for now is that this neurodevelopmental syndrome is characterized by autism spectrum disorder. Really, all of them have autism spectrum disorder. An increased head size compared to decreased head size found in Dirk-Wanné syndrome. The intellectual developmental disorder seems to be milder, with, for instance, the speech preserve. All the patients seems to have a normal language, and the facial features are really, really different from Dirk-Wanné syndrome. So to really finish this story and describe this novel neurodevelopmental disorder, so we are collecting the clinical data, we are finishing the test of the different variant effect, and we have also generated a zebrafish model for the first gain of function variant we identify to see how we will affect the head size in an animal model. So as I told you, so in the lab we are trying to better understand the clinical manifestation, to better understand variant effect, but also trying to understand not only the variant effect on Dirk-Wanné function, but to go further and to try to understand the variant effect on brain development and brain functioning. And so it's important because Dirk-Wanné is expressed during all stages of brain development and is expressed in the different cell types. So for instance, you have here a representation of some cell types really important for brain development and functioning. So first you have the neural progenitor, which can proliferate and then can be differentiated into neurons, but also into other kind of cells that would help neurons to work, which are astrocytes or oligodendrocytes. And what you can see here is like in human, Dirk-Wanné is expressed in all these different cell types, the neural progenitors, the neurons, the astrocyte or the oligodendrocyte, and really more expressed than the other protein of the same family, which are not involved in neurodevelopmental disorder. So it's a bit logical. So in the lab, we try to understand the role, particularly the role of Dirk-Wanné in neural progenitor. So these cells that can auto-renew, proliferate, and in some condition differentiate into neurons. And this neural stem cell are derived from induced pluripotent stem cell, which are also obtained for a reprogrammation of fibroblasts or blood cells. And so it was the work of Jeremy. I think it was the work he presented last year. So what he did is try to understand how Dirk-Wanné is expressed in this neural stem cell, how Dirk-Wanné interacts with protein partners. So what could be the target of in terms of protein of Dirk-Wanné in those cells. So he characterized that. And also he tried to inactivate, to do a transient inactivation of Dirk-Wanné in this cell. And he shows that a transient inactivation of Dirk-Wanné in neural progenitors will lead to a decrease of their ability to proliferate. And so this confirms the role of Dirk-Wanné in the regulation of the proliferation, the ability of neural progenitor to proliferate, and it was also consistent with what he found with a protein partner, because most of the protein partner of Dirk-Wanné are protein involved in regulation of proliferation. He also identifies that Dirk-Wanné inactivation can lead to the dysregulation of an important number of genes. And so now we wanted to go a bit further. And so because the transient inactivation was not totally sufficient to really see what we wanted to see, because it does not totally mimic what happened finally in patients with Dirk-Wanné syndrome. So we wanted to generate another model where we introduced Dirk-Wanné variants. So it was truncating variants at the beginning of the protein in iPS cells, so in pluripotent stem cell, using the CRISPR-Cas9 system. So we introduce mutations, affecting one copy or two copies of Dirk-Wanné, just to see. There is no patient with loss of function affecting the two copies of Dirk-Wanné, but because we wanted to see really the consequences of this variant, we thought that maybe generating cells with a two copy mutated could help us to really see a strong effect on brain development. And then when we obtain these cells, we differentiate them into neural stem cells. So we obtain mutant neural stem cell and then mutant neurons that we are trying to compare to the originate cells, so without the mutation, and to try to see what are the difference between the mutant and the non-mutated cells. So we have one project in the lab that try to generate neurons. So we are not passing by the neural stem cell, but directly from the iPS, and try to measure the electrical activity of this neuron and to see how DIRC1A variant could affect the neuronal activity of the neurons, their electrical activity. And in parallel, what we are doing is we are generating 3D differentiation of brain organoid, and we try to compare after how behaves the mutant brain organoid compared to a non-mutated brain organoid. And it's the work of a PhD student in the lab, Solène, who follows the work of Jeremy. So I will just briefly present what we are doing. So I cannot really give you the total conclusion because we are validating the result we obtain in another cell type in different conditions to be really sure that what we observe is directly linked to DIRC1A mutation. But I can show you how we analyze this brain organoid. So this brain organoid recapitulates some step of brain development, and so we can cut them. So it's a little sphere that we can cut, and we can see different type of cell in this slice of organoid. So I show you here a slide of organoid when you can see that you have different structures here that are really the structures that mimic the first step of brain development, one of the first step of brain development, which is the proliferation of neural progenitors. So these neural progenitors are marked here in blue. And you can see here pink cells, and these pink cells are neural progenitor which are stained with a marker of proliferation. So it means that it's neural progenitor which are highly are in active proliferation. And after in green here you have a marker of neuron cells, so differentiated neurons. So you can see here in the organoid slice how the cell can proliferate, the neural progenitor can proliferate and differentiate into neuron, which speed they take to do that, and how they do that, and if there is an increase or a decrease in proliferation of progenitors, for instance. So for now, in the first analysis we did for DIRC1A, we were able to identify some change in neural progenitor number, proliferation, and organization. And we complete this analysis by doing a whole analysis of the whole organoids by doing what we call single-cell RNA sequencing. So you take the organoid and you dissociate all the cells, and you analyze all the cells separately. So you do the transcriptome, so the expression profile of the cell, each cell separately, and after you try to combine which cell looks like which cell looks like the other one, and try to do some cluster of cells that are really similar in terms of expression. And that would mean that they are from the cell kind of cell type. They have the same cell type in brain organoid. And so after you can analyze if some cell type are decreasing or are increasing in mutant versus wild type to see how the mutation can affect this different population of neural progenitor or differentiate neuron or astrocyte or oligodendrocyte, and etc. So we also were able to identify some change in this cell composition, so meaning that DIRC1A mutation will affect brain development by affecting the cell fate, the generation of the different cell type in the brain, and we also identified some change in gene expression. So we are now doing the validation of what we found in this first iPS cell line, because we generate several iPS cell line with mutation in DIRC1A, and to be really sure that it's the effect of the mutation, we really have to analyze different different genetic background, and to really focus on the changes that are really common between these different genetic background in the mutant version compared to the non-mutated version. So we are doing that for now, and we hope to confirm the change in different genetic background, and to study after the other step will be to study how this change can occur and to study the molecular mechanism involved in this change in a cellular population in brain. And so it's what we are doing now, and as I told you, we are also focusing so for the clinical and molecular part more on the atypical variant I present you that do not lead to a loss of function of DIRC1A, to try to better understand how we could this variant could what type of neurodevelopmental syndrome is associated to this variant. And we have also another project where we focus not at the variant located at the end of the protein, but more at the beginning of the protein. And so, yeah, the idea is to improve the knowledge about all the molecular spectrum and clinical spectrum of variants in DIRC1A syndrome. Thank you for your attention. So I will thank all the team because they are really doing a great job, because it takes really a lot of time to deal with this IPS cell and doing the organoid and analyzing them. So it's really, really time consuming. Thanks also to the clinicians that are helping us to collect the clinical information, analyze them. And we have also different support. And for instance, we have a French association that support each year our work. So it's really helpful. And just to say one word, there is now a DIRC1A, I'm sure you know, they are in contact with you, a DIRC1A France association that were created a few months ago. We are preparing a day with clinician and researcher in November 2026 in Paris. And I wanted just to highlight one brother of one girl with DIRC1A syndrome in France, because he is now running, so he is doing it like this day, running between Lyon and the west of France. So it's 500 kilometers and it's 34 degrees now. So I think it's difficult, but he's doing that in order to really help to that people will know DIRC1A syndrome, to talk about DIRC1A syndrome. So you can follow him on Instagram, and also if you want to support him, there is also a barcode here. Thank you for your attention.
Thank you, Amelie, for the presentation. And it's such an important reminder that she brings up DIRC1A in France. I mean, we have partners all over, you know, in close collaboration with F1.
I'm sorry, I cannot hear you at all.
You know, but I think what Amelie's presentation.
Can you talk closer to the? Yeah, they can hear better.
So I was going to say, you know, I think the one aspect too that Amelie's presentation really highlights is really it focuses on the fact that DIRC1A is kind of like a polyglot gene, you know, and that there's all these different variants that can happen, and even in the case that she describes, where you have, you know, sort of a variant of unknown significance, you know, the person has autism spectrum disorder, like macrocephaly, that, you know, there's potentially a gain of function, you know, situation there. And it's so critical to really understand these mutations within DIRC1A syndrome, to understand what they do, what the clinical presentation is. And I think also one of the interesting aspects that I've really been kind of grappling with is, you know, when you look at diagnosis of DIRC1A syndrome, oftentimes it's easier to get diagnoses, you know, when the children are young, because, you know, they're going through these critical developmental stages and see whether, you know, milestones are being missed. And I think, you know, in the data that we've seen, and I think what we've shown, Amelie, is there's very few participants that are older. I think in your cohort that you had presented, I think maybe there was like 1 or 2 patients over the age of 30 that had a diagnosis of DIRC1A syndrome. And so I think, like, longitudinally, like, we don't really have much data in older individuals with DIRC1A syndrome. And I'm not sure if you're going to be able to hear me right now, but I would really interested to kind of get your thoughts on, you know, how you think we can maybe get more or a better understanding of older individuals with DIRC1A syndrome, because it seems like, you know, there there's a great opportunity also to understand, sort of how the brain development over time and really understand, what the presentation is in, for individuals with Dorfman syndrome. I just wonder if I can just kind of get your thoughts on that.
I'm sorry, I'm not sure I got really totally the question because it's really difficult to hear. So you mean what we need, for instance, for a clinical point of view to better characterize what would be important to know? Or maybe I missed the...
Yeah, I was just wondering like what your thoughts, but what your thoughts were about, maybe trying to understand some of the older individuals with Dorfman syndrome and, how many, maybe you've seen, or what are your thoughts in terms of, trying to understand the clinical phenotype in those individuals?
Yeah, I think it's really important. It's something that is for now a bit reduced in terms of number of patients which are actually adults. We had one in France, but it seems that finally she didn't have Dirk-Wanné syndrome because we tested this variant and it was having no effect. And so finally, it was a kind of misdiagnosis that were done. We have few other ones. For instance, in Genida, we have patient or family who fill the questionnaire for adult individual, but only a few. But we have a bit of difficulty in Genida because at the beginning, because of the French legislation, we are not allowed to ask for the mutation. So we can have the clinical information, but we cannot have the exact mutation. So we cannot ask for that, like the clinical report or the genetic report. And so I'm not totally sure that this adult, because for instance, one adult fills the questionnaire and say, fills the questionnaire so already, and say he has normal speech and blah, blah. And so I'm really not totally sure that this patient has loss of function variants in Dirk-Wanné. And so because we don't have this data, it will be really difficult to evaluate really the adult patient we have in this cohort. In France, for now, I think maybe the oldest patient are 30, something like this, but it's still very, very young. So we have a lot of difficulty to know exactly the long life trajectory of this Dirk-Wanné syndrome. I don't know in US if you have already families with really child with more than 45 years old or something like this, but we do not have a lot. Yes.
Does anybody?
Sorry.
Yeah. Does anybody have any questions for Amelie that they want to ask?
So the app is considered a 25. Is that what she's looking for? Someone older?
Yeah, I think she was referring to just older individuals. Yeah. And then I'm sort of curious too, in France, do you have any familial cases of Dirk-Wanné syndrome?
So, sorry, you mean transmitted like transmitted inheritance, like siblings or like one parent and one child?
Yes.
Yeah, no. For now, all the variants we have are de novo. Even the variants presumed to be gain of function, so that will lead to a milder form of neurodevelopmental disorder. What we thought it would be mild. For now, we do not have any familial cases.
Yeah, the question from the audience here, is there any information, from the parents and the patients here, any information that they can provide to you or information be shared to help you in your research?
Yes, of course. So if family want to fill the Genida questionnaire, it will be really helpful. They also, because we are not allowed to ask for the clinical report, but there is like a field where you can put comment and you can put the mutation here, for instance, of course. And so this will be really helpful. We have also we are also collecting MRI data, for instance. So for now, we began with French families because it was easier. But I don't know how it could be easier to transfer this kind of information from abroad. So I don't know, but it could be something. If you are aware of variants that really are this kind of atypical variant, so it means that variants found in a patient with neurodevelopmental disorder, but which really do not look like the other individual with Dirk-One syndrome, it could be something really helpful to help to better characterize this novel genetic form. And I think like both will inform, so it will be also important for Dirk-One syndrome, because if we know what too much Dirk-One or too less Dirk-One is doing, it's important for both of this form. Yes, so it's the things that could be interesting. I saw also sometime like in the Facebook group, something like this, that they are familiar with variant of uncertain significance. For those one, I would really maybe advise to really discuss with the geneticist or the neurologist to try to see how they can go further. For instance, now we have the methylation signature, so it's something that could be easy, not to stay with this uncertain significance variant, but really to have an answer if it's deleterious, if it's involved or not. I think it could be really, so there is this episignature, it's quite of convenience. I don't know how it works in US if you want to be to pay for this kind of tests, if you have some insurance to help with that. But yeah, it can really help to have the answer for this variant of uncertain significance.
Yeah, and I think that's a really important point because, you know, oftentimes like when you get a diagnosis, even of Dirk-One syndrome, or you don't, you just have a variant within the Dirk-One gene. You know, sometimes like you can get information from the geneticist and, you know, let's say your mutation's like 956 TA or something like that. Oftentimes, like that's almost like the end of the road if they call it a variant of unknown significance, meaning like they don't know if it's pathologically, you know, responsible for Dirk-One syndrome. And so I think it's challenging even in the US to then take that next step further to say, okay, what does this actually do? And I think, you know, from what Amelie presented about, you know, doing the epigenetic signature or looking at the kinase function of the variant and kind of doing those, you know, translational science assays. That's not really something that's super common and actually easy to come by in the US. So, you know, I do think that would be a great opportunity to potentially, you know, collaborate on those and get that information, especially in those cases of variants of unknown significance, because oftentimes we have to be proactive and sometimes taking that step to understand what is this mutation actually.
And I think I gave my email at the beginning of the presentation, but do not hesitate. Yeah, if there is a family which were given a genetic report where it's written variant of uncertain significance, do not hesitate to write me an email and we can see yeah, maybe how we could interpret it or how we can organize something with the Canadian lab, for instance, if it's not possible to do the episignature in US or but yeah, do not hesitate. It will be a pleasure for me to try to see if this variant could be or not involved.
Okay, awesome. Well, I think we're just a tad behind on that, but we'll go ahead and take a short break, maybe for about 10 minutes or so, and then come back, I think, for Craig's talk. So, all right, well, thanks. Let's take a break and see you in a little bit.
Thank you.
Thank you.
I will try to stay maybe for the Craig talk, but I can stay on this panel or I have to reconnect in the other?
You can just stay here.
Okay, perfect.
Thank you.